Hello everyone and welcome back to another physiology video on sqadia.com. Remember that in our last video we talked about cell structures and its organelles. So today we're going to learn some details of cell functions. So let's get started. But before that, let us revise the cell and its organelles first. Now this is a typical diagram of the cell, and here's the cell membrane in which all the organelles are enclosed. Here's the nucleus, and inside it is the DNA. Now here we have the cytoplasm. It is the fluid of the cell where all the organelles are suspended. Here's mitochondria. Then we have ribosomes floating over here. It is endoplasmic reticulum, and there are two types of endoplasmic reticulums as we discussed before: smooth endoplasmic reticulum, which is without ribosomes, and the rough endoplasmic reticulum, which is with ribosomes. Then we have Golgi apparatus, and here are the centrioles. Lastly, this is lysosomes. So we will be talking mainly about the ingestion process, which is endocytosis, and it will be associated with lysosomes. And then we're going to talk about endoplasmic reticulum, Golgi apparatus, and of course, how can we forget the powerhouse of the cell, which is mitochondria? And we're going to discuss all of their functions. So let's get started with endocytosis.
Thank you so much for the recap and a short upcoming glance. Let me take it from here. So, what is endocytosis? It is basically derived from two words. Endo, which means inside. Cytosis, which means related to cell. So, endocytosis means inside the cell. Endocytosis occurs in two ways. One is pinocytosis. The other is phagocytosis. Pinocytosis is the ingestion of small particles, while phagocytosis means the ingestion of large molecules. Both of these processes are almost the same. Pinocytosis means the ingestion of small particles that form vesicles of extracellular fluid and particulate constituents inside the cell cytoplasm. Pinocytosis occurs continually in the cell membranes of most cells, but is especially rapid in some cells. Fun fact: did you know that pinocytosis occurs so rapidly in macrophages that about 3% of the total macrophage membrane is engulfed in the form of vesicles each minute? Pretty fascinating. Let us look at the whole process. So here is the cell membrane. What will it do if it wants to ingest a protein or a foreign molecule? Well, for that, let's zoom in a bit and see what is happening. All the cell membranes have specific receptors that attach to a specific protein or foreign molecule. Let's say some proteins come to the cell membrane and attach themselves to the receptor. Now the cell will activate itself to invaginates or make some small pits. They are known as coated pits. Now coated pits have some fibrillar proteins inside, and we name them clathrin, and some other molecules are also there along with filaments of actin and myosin. Now the pit invaginates inwards to make sort of a mouth that is eating that protein. An invaginated portion of the membrane breaks away from the surface of the cell, forming a pinocytotic vesicle inside the cytoplasm of the cell. Pinocytotic vesicles are so small, usually only 100 to 200 nanometers in diameter, that most of them can be seen only with an electron microscope. Now how this invagination occurs and how the clathrin gathers around this protein, these mechanisms are still unclear, but one thing is for sure: all of it is aided by the energy which is provided in the form of ATP. This process also requires the presence of calcium ions in the extracellular fluid, which probably react with contractile protein filaments beneath the coated pits to provide the force for pinching the vesicles away from the cell membrane. So, they are sort of aiding the process of pinocytosis. let us come towards phagocytosis. Phagocytosis means the ingestion of large particles, such as bacteria, whole cells, or portions of degenerating tissue. Phagocytosis occurs in much the same way as pinocytosis, except that it involves large particles rather than molecules. Phagocytosis starts when a particle such as a bacterium, dead cell, or tissue debris binds with receptors on the surface of the phagocyte. The process is the same as pinocytosis, but let us revise it a bit. First, the cell membrane receptors attach to the surface ligands of the particle. Then, the edges of the membrane around the points of attachment evaginate outward within a fraction of a second to surround the entire article. After that, actin and other contractile fibrils in the cytoplasm surround the phagocytic vesicle and contract around its outer edge, pushing the vesicle to the interior. Lastly, the contractile proteins then pinch the stem of the vesicle so completely that the vesicle separates from the cell membrane, leaving the vesicle in the cell interior in the same way that pinocytotic vesicles are formed. Now at the start, we mentioned that lysosomes play an important role in endocytosis and its related processes. So, what it does is that the particles that were ingested through the process of pinocytosis and phagocytosis digest those substances. So let us continue the process from here. Here are the pinocytotic and phagocytotic vesicles. Now some lysosomes will come towards them and get attached to the vesicles. They release enzymes known as hydrolases in them. A digestive vesicle is formed inside the cell cytoplasm in which the vesicular hydrolases begin hydrolyzing the proteins, carbohydrates, lipids, and other substances in the vesicle. The products of digestion are small molecules of substances such as amino acids, glucose, and phosphates that can diffuse through the membrane of the vesicle into the cytoplasm. What is left of the digestive vesicle, called the residual body, represents indigestible substances. In most cases, the residual body is finally excreted through the cell membrane by a process called exocytosis, which is essentially the opposite of endocytosis. Thus, the pinocytotic and phagocytic vesicles containing lysosomes can be called the digestive organs of the cells. Another special role of the lysosomes is the removal of damaged cells or damaged portions of cells from tissues. Damage to the cell caused by heat, cold, trauma, chemicals, or any other factor induces lysosomes to rupture. The released hydrolases immediately begin to digest the surrounding organic substances. If the damage is slight, only a portion of the cell is removed, and the cell is then repaired. If the damage is severe, the entire cell is digested, a process called autolysis. In this way, the cell is completely removed, and a new cell of the same type is formed, ordinarily by mitotic reproduction of an adjacent cell to take the place of the old one. The lysosomes also contain bactericidal agents that can kill phagocytized bacteria before they cause cellular damage. These agents include the following: lysozyme, which dissolves the bacterial cell wall; lysoferrin, which binds iron and other substances before they can promote bacterial growth, and acid at a pH of about 5.0, which activates the hydrolases and inactivates bacterial metabolic systems. That was all about endocytosis. Let us discuss further functions next.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.